This is Dr. Shanmugapriya for the Speed Learning app, and I'm here to discuss about recombinant DNA technology and blotting techniques. And why do you have to know about all these techniques? Answer is very simple. We often get asked about all these techniques, right? These days there is a shift. There's a shift from theory-based question to application-based questions. So there are many questions which are asked about recombinant DNA technology and about blotting. First, about recombinant DNA technology, it is one of cloning techniques. So what is cloning? Cloning means production of identical copies. And here we are trying to produce identical copies of DNA. Why will we want to produce identical copies of DNA? Does it have any application? Yes. If you want to produce a particular protein on a large scale, the first raw material that you want is the concerned gene should be copied multiple times. So only when you have multiple copies of that gene, you can expect multiple copies of mRNA. Only then you will get multiple protein products. So it has got genetic engineering applications. Okay. Next, it has got forensic application. Because from the crime site, maximally you get a hair follicle or a blood spot, right? In either case, you will be able to extract 1 or 2 copies of DNA. So before you can perform any DNA fingerprinting techniques, you have to amplify that DNA multiple times. So production of identical copies of DNA has got many applications, but if you look at the process, it is nothing but replication of DNA. And for replicating DNA, we have few requirements, right? The first requirement is you need the enzyme which replicates. That is, you need a DNA polymerase enzyme. And then there are requirements for the DNA polymerase. All polymerases need primers. You need deoxynucleotide triphosphate. You need magnesium or manganese to act as a catalyst. And then you need a buffer. So all these requirements I've already told you when I discussed about replication. So these requirements, you need something to satisfy these requirements. To satisfy these requirements, if you are dependent on a whole living cell like E. coli, to meet all these requirements of replication, if you are dependent on a whole living cell like E. coli, you call it as a cell-based cloning. And this cell-based cloning is nothing but the recombinant DNA technology which we are trying to discuss now. So what is recombinant DNA technology? It is otherwise called as cell-based cloning, wherein all the requirements of replication are satisfied by a living cell like E. coli. Now, what are the steps of recombinant DNA technology? For understanding the steps of recombinant DNA technology, I want you to imagine that you are involved in producing insulin on a large scale. So, to produce insulin on a large scale, as you all know, you need insulin gene. From where do you get insulin gene? From human chromosome. So, the first step is you take human chromosome. The first step is you take human chromosome and you cleave this human chromosome using a DNA scissor called as restriction endonuclease. So, what is the first tool that you want for recombinant DNA technology? It is restriction endonuclease. What do you use this for? You use this enzyme to cut human chromosome into multiple fragments. And if you ask me why should I cut human chromosome into multiple fragments, the answer is after you cut it into multiple fragments, out of all the fragments, you're going to identify the fragment with the gene of interest and you're going to amplify just that fragment. That's optimal, right? Instead, if you try to amplify the whole human chromosome, it's a waste of time and waste of raw material. So, the first step is you take human chromosome, you cleave human chromosome into multiple fragments using a DNA scissor called as restriction endonuclease. So, these are the fragments you have obtained. Now, in the next step, out of all these fragments, you are going to identify the fragment with the gene of interest. And to identify the fragment with the gene of interest, we employ a technique called as hybridization technique or blotting technique. So what is the second tool you want for recombinant DNA technology? It is hybridization technique or blotting technique. We'll be discussing about that in detail probably in another 15 minutes. For now, remember this is the second tool that you want. Now, using this tool, you have identified that this is the fragment which has got the gene of interest. What do we do now? We attach this fragment with gene of interest to a vector. So, what do you think is the role of this vector? This vector, as the name indicates, acts as a vehicle. And it carries the fragment with gene of interest and it gets into E. coli. So, what is the role of vector? Vector is basically a vehicle which carries the fragment with gene of interest. Okay. So, what do? We take a vector, we cut the vector and then we insert the fragment with gene of interest to form something called as recombinant vector. Now, do you understand the reason behind the name recombinant DNA technology? We call it as recombinant DNA technology because we form a recombinant product involving a vector and eukaryotic gene fragment. Okay. After you have formed the recombinant vector, now comes a difficult step of recombinant DNA technology where you are trying to introduce this recombinant vector into E. coli. This is not easy because E. coli is a prokaryote and prokaryotic cells have cell wall. And introducing anything across cell wall is not that easy. So, what we do here is using high electric currents, we introduce pores onto the cell wall and through these pores, recombinant vectors will be taken up. So, this step is called as electroporation step. Okay, and this electroporation step is considered as the rate limiting step of recombinant DNA technology. Why? Because the success of this technology depends upon, or the number of products you get through this technology depends upon how efficiently you have introduced recombinant vector into E. coli. If you have introduced many recombinant vectors into E. coli, you will get more products. If you have not introduced recombinant vector into E. coli, you are not going to get the product. So, which is the rate limiting step of recombinant DNA technology? It is electroporation step or the step where you introduce recombinant vector into E. coli. And once you are done with this step, the rest E. coli takes care. Because whenever the vector replicates, you get multiple copies. Yeah, whenever the vector replicates, you get multiple copies. And whenever the cell divides also, you get multiple copies. Yeah, so this is an outline of steps involved in recombinant DNA technology. So, what are the steps involved in recombinant DNA technology? First step, you take human chromosome. You cleave human chromosome using a DNA scissor called as restriction endonuclease. Next, out of all the fragments, you find out the fragment with the gene of interest using a technique called as hybridization technique or blotting technique. After you identify the fragment with gene of interest, you have to attach it to a vector. So, what is the third tool you want for recombinant DNA technology? It is a vector. So, you attach the fragment with gene of interest and the vector to form recombinant vector, hence the name recombinant DNA technology. Now, you have to introduce this recombinant vector into E. coli, which is not that easy because E. coli is a prokaryote with cell wall. So, using high electric currents, you introduce pores onto the cell wall and through those pores, recombinant vectors get taken up. So, we call that step as electroporation step. And that's the rate limiting step of recombinant DNA technology. Once you are done with this step, the rest E. coli takes care. Whenever the vector replicates, you get multiple copies. Whenever the cell divides also, you get multiple copies. So, tell me what are the three tools of recombinant DNA technology? So, the first tool is restriction endonuclease, okay, which is used for cutting human chromosome and for cutting the vector. Second tool is hybridization technique or blotting technique. And the third tool is vector. So, let's discuss about these tools one by one. So first about restriction endonucleases. So what are restriction endonucleases? As the name indicates, these are enzymes which cut at restricted sites or they cut at specific sites. And those sites are generally palindromic sequences. So what is palindrome in literature? What does palindrome actually mean? Palindrome in literature means a word or a phrase which carries the same meaning when read from either side. Right? For example, rotor, R-O-T-O-R, read it from either side, it's the same. Yeah, so that's a palindrome. Now, what is a palindromic sequence in genetics? In genetics, palindromic sequence is a double-stranded sequence wherein the 3' 5' strand sequence is the same as 5' 3' strand sequence read in the reverse direction. Do you understand this? Palindromic sequence is a double-stranded sequence wherein the 3' 5' strand sequence is the same as 5' 3' strand sequence read in the reverse direction. For example, if 3' 5' strand sequence is GGATCC, okay, tell me what will be the complementary sequences for this? It is CCTAGG. So don't you see that the 3' 5' strand sequence is nothing but the 5' 3' strand sequence read in the reverse direction. So this is a palindromic sequence. Now, depending upon how a restriction endonuclease cuts this palindromic sequence, we have two types of restriction endonucleases. One is a sticky end producing restriction endonuclease. The other one is a blunt end producing restriction endonuclease. So, what is a sticky end producing restriction endonuclease? In this case, the enzyme cuts both the strands at two different sites. Instead of cutting both the strands after the first nucleotide, cutting both the strands after the second or after the third, what does this enzyme do? This enzyme cuts both the strands at two different sites. For example, it cuts the top strand after the first nucleotide and it cuts the bottom strand only after the third nucleotide. In that case, the cut ends produced are staggered. Yeah, this will be one end and this becomes the other end. So, the cut ends produced are staggered. Yeah, in that case, you call it as a sticky end producing restriction endonuclease because suppose this represents the end of the fragment with gene of interest. Yeah, this represents the end of fragment with gene of interest and this represents the end of the vector. Yeah, you know, using the same enzyme, you would have also cut the vector. Why do you have to cut the vector? Only after you cut the vector, you can introduce the fragment with gene of interest. Okay. Suppose this represents the end of the fragment with gene of interest and this represents the vector. What happens when you bring them close together? Because they have hanging edges. Yeah, they stick together easily. They have hanging edges with complementary bases, so they stick together easily. So, what is the advantage of using a sticky end producing restriction endonuclease? Forming recombinant vector is easier. Otherwise, you know or do you understand that forming recombinant vector is difficult? Yeah, because you are linking two unrelated genomes. Yeah, one is a prokaryotic vector, the other one is eukaryotic gene fragment. So, formation of recombinant vector is not that easy. So, if you use a sticky end producing restriction endonuclease, formation of recombinant vector is easier because the edges are hanging and they have complementary bases. So, what are blunt end producing restriction endonucleases? In this case, the enzyme cuts both the strands at the same point after the first or after the second or after the third. In that case, there are no hanging edges, there are no complementary bases. So, even if you bring the fragment with gene of interest and the vector, they don't stick together easily. That is why they are called as blunt end producing restriction endonucleases. And these days, we use mostly sticky end producing restriction endonucleases, and the only blunt end producing restriction endonuclease which is given as an example in Harper is HPA1. Yeah, there is a tabular column in Harper which is telling you about all these restriction endonucleases. The first column tells you the name of the enzyme and the second column tells you about the restriction site at which it cuts, and then it tells you whether it's a blunt end or a sticky end producing enzyme. And if you look at the tabular column, the only one example that is given for blunt end producing restriction endonuclease is HPA1. So that is all you need to know about the first tool, which is restriction endonucleases. Don't get confused about other types. There are other classifications saying type 1, 2, 3, 4 and 5. Those are all of not much of practical importance. So if you know sticky end and blunt end types, that's enough. Okay. Now with this enzyme, you have cut human chromosome into multiple fragments. What is the next step? Of all the fragments, you are going to identify the fragment with the gene of interest. So how do you do it? Using hybridization technique or blotting technique. So what is the first step of hybridization technique or blotting technique? You have to separate all the fragments based on length by electrophoresis. So the first step of blotting technique is electrophoretic separation of all the fragments. So what I have drawn here is a gel. Yeah, this is a gel and all these are wells. There are 3 wells. So into one of these wells, you have added the fragments. You have added all the fragments which you have obtained by cutting the human chromosome into multiple fragments. Now you apply electric current. Close to the point of application is connected to negatively charged electrode. The opposite side is connected to the positively charged electrode. Now you switch on the electric current because all your DNA fragments are negatively charged. They start moving towards positively charged electrode. Yeah, they start moving. What do you think will be the determining factor of the speed here? Yeah, everything else is similar. The only differentiating factor will be length. Longer fragments cannot move faster, so they stay close to the point of application. Shorter fragments move faster. So what is the first step of hybridization or blotting technique? You separate all the fragments based on length by electrophoresis. The next step is you denature all the fragments. Why do you denature all the fragments? Because in the next few steps, you are going to use a probe. Yeah, and that probe is going to be single-stranded and it is going to be complementary to the gene of interest. And for that probe to hybridize, these fragments should be single-stranded. So after separating them by electrophoresis, you denature all the fragments. After you denature the fragments on the gel, you take the gel and you blot it on a nitrocellulose paper. So the third step is blotting step. What do you do in blotting step? You blot the gel on a nitrocellulose paper because handling gel is very difficult. Until your bands are within the gel, they will not hybridize with probes. So you take the gel and you blot it on a nitrocellulose paper. That is why this technique is called as blotting technique. And then you take the paper. So now it's no more in the gel. It is all in the paper. Now you take this nitrocellulose paper and you dip this nitrocellulose paper in a solution. Yeah, you dip this nitrocellulose paper in a solution containing radiolabeled probe. So have you seen a petri dish? You must have seen it many a times, right? So you take a petri dish, you fill the petri dish with a solution containing radiolabeled probe, and into this petri dish you drop the nitrocellulose paper. So these radiolabeled probes, as I've already told you, are labeled with radioactive substances. They are single-stranded and they are complementary to the gene of interest. So these radiolabeled probes will try to find complementary sequences on all these bands, and once they find complementary sequences, they hybridize. Okay, suppose they have found their complementary sequence on the third band, they go and hybridize here. So what is this step called as? This step is called as hybridization step. Why do you call it as hybridization step? Because in this step, the probe hybridizes with the gene of interest, and that's why the technique is called as hybridization technique. So after you give enough time for hybridization, you take out the paper and you wash away excess unbound probe. Okay, and then you expose the paper onto a X-ray. You expose the paper onto an X-ray. This is called as autoradiogram. Okay, and because it's radio-labeled, it will form a marking on an X-ray. So by looking at the X-ray, you will know which band has got the fragment with gene of interest. So this is called as hybridization technique or blotting technique. So what are the steps involved in hybridization or blotting technique? The first step is you separate all the fragments based on length by electrophoresis. Next step, you denature all the fragments so that the probe will be able to hybridize. So second step is denaturation. Third step is you take the gel and you blot it on a nitrocellulose paper. So this is called as blotting step. And then you dip the nitrocellulose paper in a solution containing radio-labeled probe. So these radio-labeled probes try to find complementary sequences on all these bands. Once they find complementary sequences, they hybridize. So it's a hybridization step. After you give enough time for hybridization, you wash away the excess unbound probe and you expose it to X-ray. Yeah, that's called as autoradiogram step. And by visualizing the autoradiogram, you will know which band has got the gene of interest. Now that you have found out that the third band has got the gene of interest, can you use the third band for cloning? Can you use it for recombinant DNA technology? You can't, because what have you done to it? You have disturbed it so much. Yeah, what did you do first? First you denatured it, so it's no more double-stranded, single-stranded. Then you allowed it to hybridize with the radio-labeled probe. And then you have exposed it to an X-ray. So what you are looking at is just a marking on the X-ray. So that mark on the X-ray will just be used as a guidance for you to find out which band has got the gene of interest. So what are you supposed to do? You are supposed to repeat the whole process all over again. What will you do? You take the same human chromosome. You cleave the human chromosome with the same restriction endonuclease. And then you separate all the fragments based on length by electrophoresis using the same tank, same power supply, same power pack, same settings, same buffer. So you'll have to exactly replicate what you've already done. And after separation, now you know the third band has got the gene of interest. Don't disturb it. You just elute the third band and use it for cloning. Is that clear? And this technique is called as southern blotting. This technique is called as southern blotting because it's a technique which we have used to find out an unknown DNA fragment. And it is named after the person who invented this technique. If you are using the same principle, but for identifying an unknown RNA fragment, that will be called as northern blot. If you are using the same technique, but for identifying unknown RNA fragment, that will be called as northern blot. So what do you think will be the difference between southern blot and northern blot?